So our next speaker is Peter Kim, and Peter is the Ludwig Professor of Biochemistry here at Stanford. His current research is focused on devising new strategies to enable vaccine development. He was president of Merck Research Labs from 2003 to 2013 and oversaw the development of more than 20 new medicines and vaccines in that position. I could say a lot more about Peter now, and he wouldn't be happy. So I will simply say the title of his talk is New Frontiers for Vaccines Against Infectious Disease. Peter.
Thank you, Lucy. It's a pleasure to be here at the symposium, and I'd like to describe the work that we're doing to try and develop vaccines against infectious disease with a focus on pandemic preparedness. Here's the outline of what I'd like to talk about. Just in two or three slides, talk about a major initiative of the lab, and that is to try to work on what I call immunofocusing, and then tell you two stories, one very quickly on Ebola, but then I'd like to spend most of my time focused on influenza. The problem statement for immunofocusing is simple. There are monoclonal antibodies, several of which have been approved for use in people, and many more that have been shown in animals to prevent infectious disease. And for those monoclonal antibodies, we know exactly where they bind, and we know exactly what epitopes they're targeting. But monoclonal antibodies are not an efficient way to prevent disease as compared to vaccines. But we know what we need to do. The monoclonals tell us what we need to do. We need to come up with a vaccine that elicits antibodies that target that specific epitope. But we can't do that. And that's perhaps most clearly demonstrated in the 40-year effort to try and develop an HIV vaccine, which has proved to be, so far, not possible, even though we have broadly neutralizing antibodies that have been extraordinarily well characterized that can prevent HIV infection, as shown recently in a clinical trial conducted in people over a period of 80 months. So we know what we need to do, but we can't do it with a vaccine. So that's led to this concept of immunofocusing, where we know what the target is, but now we have to figure out how do we make vaccines that actually elicit antibodies that hit that target. And that's what we're referring to as immunofocusing. I won't talk about this more, but that's a major effort of our research. So I'll tell you one very quick story about how we've tried to do immunofocusing to try and come up with universal and accessible Ebola vaccines. Ebola virus has caused outbreaks. Here are the outbreaks that are shown. There are 3 different species of Ebola that have led to serious outbreaks in humans, and this continues today. And there is one Ebola vaccine that's been approved, and that's a vaccine that targets the species Ebola Zaire. But that vaccine is species-specific. It doesn't work against the other Ebola species, and it requires low temperature, minus 80-degree freezing. And so as you're trying to deal with a vaccine in warm climates such as Africa, it's very troubling to have to keep it at minus 80 degrees C. As a result, this vaccine has been used only for what's called ring vaccination, and that's to vaccinate people that have been either are known to have contacted or are suspected to have contacted somebody who's been infected. And the problem is shown here with the red circles on the molecule of the glycoprotein, in that those parts of the glycoprotein are highly variable, and that leads to the species specificity. So what we've done is to try to immunofocus by adding hyperglycosylation, by adding sites where we can glycosylate the protein and thereby shield the regions that are variable. And without going into details of what we did, we ended up adding 3 glycans in this, what's shown in green here, in the GP plus 3, and this is on the backbone of a ferritin nanoparticle, which we use to improve the immunogenicity. And we ended up with a vaccine candidate that we tested in mice. And we took that serum from that candidate and asked our colleagues at BU to test it in a BSL-4 facility against authentic Ebola Zaire virus, and indeed the GP+3 shown in green shows the best activity of the constructs that we made. But most importantly, when we tested the GP+3 ferritin construct in mice and obtained the antisera, what we found was the antisera could neutralize all 3 species of Ebola virus. So we now have, for the first time, a single vaccine that can actually lead to neutralization not just of Zaire, but also Bundibugyo and the Sudan Ebola virus. Also, we've shown that this particle is stable at 37 degrees C for at least 2 weeks. And so we have something which potentially is a room temperature stable vaccine that could be used in these sorts of settings. And so this is a work in progress, and we're hopefully now going to move forward to try and develop this. But I'd like to move on to influenza. And the issue with influenza, as many of you know, is that the vaccines, as well as the infection, lead to a very biased antibody response. The overwhelming response of the antibodies is against what's called the head region. And the head region, as shown in the red here, is the part of the protein that's highly variable. It's what changes year to year, and that's why every year we need to have a new vaccine made so that we can elicit antibodies that are specific against that particular strain at the head. There are rarely antibodies that are made against the stem, and some of these antibodies target very conserved regions, but they're rare. Nonetheless, workers have been able to isolate such antibodies, and some of these antibodies are incredibly broad and can neutralize influenza strains, both from what's called group 1 as well as group 2 sequences. And so what we want to do with a vaccine, we know, is to target that conserved stem, and in particular those conserved epitopes that we know lead to broadly neutralizing antibodies. And this has been work that's been going on in the field for many, many years. People are really putting a lot of effort into trying to figure out how to immunofocus toward the stem. Well, we wanted to test the hypothesis that perhaps the reason why it's so hard to elicit stem antibodies is that the stem is actually sterically hindered on the surface of the virus or on the surface of vaccines that are created from virions. And this shows an electron tomography picture of a flu virus. And you can see that the heads are well accessible and separated, but from the side you can see that the stems are actually pretty close packed. And it's known that antibodies can still bind to the stem region, as I showed you on the previous slide, but it's also known that the affinity for those antibodies for the stem region on intact virions is about 2- or 3-fold lower than the affinity of a freely exposed hemagglutinin. So there is some hindrance of an antibody getting in there. Our hypothesis was that for a big B cell that needs to get in there, the hindrance would be much larger than for a small antibody. And so we wanted to increase the accessibility of the stem. And our thought was that what we needed to do was to figure out how to turn this hemagglutinin protein upside down and have the stem be more exposed. Our approach was to use a variation of a method that was developed by Darrell Irvine at MIT, where he showed that if you chemically conjugate phosphoserine peptides to an antigen, you can then have very tight association with a adjuvant that's called alum, or aluminum hydroxide, which is an adjuvant that's been used for over 90 years. This involves chemical conjugation, and so what Duo Xu, a former postdoc in the lab, did was to ask whether or not it could be replaced with a string of negatively charged aspartic acids, which could be genetically encoded into the protein without having the need for chemical conjugation. I won't show you the data, but it works. It works beautifully as a method to improve the immunogenicity. But then what he did was he asked whether or not he could find a region in the head, a residue in the head, where he could insert a loop of aspartic acid residues. So he could insert a loop of negatively charged amino acids, and since the hemagglutinin protein is a trimer, a homotrimer, once he found such a position, he could then have a tripod, as shown on the bottom here, and that tripod would stick to alum and have the head stuck on tightly to alum and the stem exposed. And he showed that that indeed was the orientation. I won't show you that data. But when he immunized mice with either the construct which is called H2HA, which is just normal hemagglutinin, or his reoriented H2HA, his upside-down HA, which is called Rho-H2HA. What he saw, each of these circles is the antisera from an individual mouse. And what he saw was that the mice that were immunized with the upside-down H2HA that started with H2 cross-reacted not just with H1 and H5 hemagglutinins, but they also cross-reacted into what's called group 2, the H3 and the H7 hemagglutinins. So in the red circles, we have animals that are producing antisera after immunization with the upside-down HA that bind to all of the different HAs that were tested. And so basically a so-called universal approach. Then, together with our colleagues Christopher Barnes and his student Josh Carter, we used a method called MPEM to look using the electron microscope at the antisera and where they bind. And when you use just the antisera following immunization with normal H2HA, as shown in the middle, and you ask whether or not you get binding to H2HA, you see that it binds all over the place. But when you ask for binding to H3 or H7, you don't see any binding. In contrast, when you take the antisera you obtained with the upside-down HA, Rho-H2HA on the right, once again, when you use H2, you see binding all over the place, but now when you ask for binding to H3 or H7, you see specific binding in the stem region at the region that we wanted to target. So we're getting stem-specific responses to the hemagglutinins that are cross-reactive. So now turning to the current issue. As you know, we've had influenza pandemics in the past century. The 1918 pandemic is estimated to have killed over 50 million people, and the world was a lot smaller then than it is today. So this is what we're concerned about. The pandemics arise when the influenza virus reassorts. Influenza virus has a segmented genome, and so if a person happens to be co-infected with the seasonal flu, a human flu, but also, for example, an avian flu, at the same time, there's a potential for the virus to reassort and produce a potential pandemic strain. And as you are all aware, earlier this year, there was the first infection of a human with the H5N1, and in particular with what's referred to as a highly pathogenic avian influenza A virus, which has been having a very devastating effect on some bird populations, the first time a dairy farm worker in Texas was infected. And most recently, there's been a study of this virus, and although the virus did not cause a serious illness in this farm worker, it's been shown that that virus actually can replicate in primary human alveolar epithelial cells, and also that that virus is lethal in mice and ferrets, ferrets being the model system that is used very extensively in influenza virus research for both prevention and transmission. And just today, in Science, it was shown that a single mutation in this virus, in this Texas virus, actually switches the specificity. The Texas virus actually has a specificity for the bird type sialic acid, but a single amino acid change can switch it over so it now recognizes the human type. So this is the concern. And as a result, there's been efforts to try to stockpile existing H5N1 vaccines. And in the United States, there are 3 H5N1 vaccines that have been approved. They're indicated here. But they were approved based on the strains that came out in 2004 and 2005. And if you ask, where are the differences in the sequences for the hemagglutinins for these 3 vaccines that have been approved for H5N1 versus the Texas strain in that person, what you see is, as you would expect, the head region now has been highly mutated between 2004 and today. So if the current vaccines that have been approved actually have heads that are quite different than the head region of the Texas strain, whereas you can see that the stem region down at the bottom is still highly conserved. The CDC has responded to this by nominating some pre-pandemic vaccine candidates that bring the H5 strains up to more recent strains, the 2020 version and the 2021. And fortunately, as you can see on the right here, that leads to strains that have that are closer to the Texas strain. But we're in this sort of dilemma where we don't know if we're going to have an outbreak, and if we do have an outbreak, we don't know when it's going to occur. And so in the meantime, we're going to be constantly playing catch-up to try and stay current with something that's closer to the head of what we're worried about. So in unpublished work, we asked whether or not we could do a similar thing with H5 and found a residue where we could again turn it upside down, and so turn the virus, the hemagglutinin, upside down. And here I'll pause and actually show you some of the data that shows that we have turned it upside down. On the top is just the normal, is the Rio HA that's been biotinylated at its C terminus. And so there's no alum. And we put streptavidin down on the plate, on the ELISA plate, and we bind the hemagglutinin to that plate so that it's now just in solution, bound by its C terminus. And then we ask whether or not the 5 antibodies shown on the right bind to that ELISA plate. And as you can see, all 5 antibodies show good binding to the different epitopes. So we're recognizing all the epitopes. But then to measure the ELISA binding on alum, what we do is we put down a protein, in this case is a green fluorescent protein, that has the aspartic acid linkers on it already. So that's just to put something down on the plate that has the ability to bind alum. Then we add the alum, so that it binds to the alum. And now we come in with the Rio HA, so that it binds, we think, in upside down with a tripod. Now when you ask for the binding of the antibodies, what you see is that the red antibody and the blue antibody no longer bind to the hemagglutinin. So the head epitopes are shielded because the protein is upside down and stuck on alum. The one antibody that binds to the side in yellow still binds, and the 2 stem-binding antibodies still bind very well. So we think we really have turned this molecule upside down. And then when we ask about what sort of response we get in mice, what you see is that we get a very good response, as shown in the middle, against the Vietnam H5 hemagglutinin. But we also now cross-react with the avian flu from 2020 from Hubei and the 2023 from Bangladesh. We get very good binding to those molecules as well. But importantly, we also get good binding to all of the other HAs that we tested, including both group 1 and group 2. So one of the challenges is how to develop such a molecule and the timeline for doing that. But what we think is that this presents an opportunity, actually, for potentially developing this as an add-on to existing seasonal flu vaccines, which perhaps could give, for example, additional protection against H1, and then actually have that be a vaccine that could be used to, as part of the seasonal vaccination, so that in the event of an outbreak, a person would have some pre-existing H5 antibodies against the stem. And because the antibodies are against the stem, what we think is that this would avoid the variant chasing problem that we are all so very familiar with. So in conclusion, we have a single HA-based immunogen with a very safe adjuvant. Alum has been used for over 90 years and is used in infants in the United States. There are 3 vaccines that are given to infants with alum formulations. It elicits a broad cross-reactive immune response that is focused on the stem region. And this approach is orthogonal to the existing approaches that people are taking to try and develop or prepare for pandemic H5 vaccines. And what we hope is that because we're targeting the stem, and because the stem is highly conserved, we can avoid the variant chasing strategy that's going to be with us with the traditional vaccine approaches to influenza in the event of an outbreak. And so with that, I'd just like to acknowledge the people who have done this work. I want to give a big shout out to Duo Xu, who was just a tremendous postdoc who's now just started as an assistant professor at the University of Wisconsin in Madison. We've collaborated with Christopher Barnes on the EM studies, and with Bali Palendran on the T cell responses, and with our collaborators at Boston University on the BSL-4 work. And I'd like to thank you for your attention. Be happy to answer questions.
Thank you, Mr. Kim. I'm Greg Martin. So when talking about vaccines, so basically there's vaccine development that's made for humans. So I was wondering, in the, say the H5N1 is being spread from animals and birds to humans. So can you tell me what level are animals and birds given any kind of vaccines to try to prevent them from getting sick?
Yeah, thank you. I'm actually, Kanta would be a much better person to answer that question, so why don't I let you answer that?
Yeah, so it is, it's possible to prevent disease in poultry with vaccination, but it was controversial for many years because once an animal is immune, whether they're immune because of infection or vaccination, it has consequences on export and trade. So many countries actually opted instead to cull infected birds. Now there's a recognition that that's not feasible, that we've got a panzootic, and I'll be talking about that a little more. And so the WHO and the World Organization for Animal Health have come out with a statement that actually does encourage the use of vaccination, and have basically lobbied for a change in consequences for trade, a recognition that we really ought to be using vaccination as an additional measure to control disease in animals. The issue with poultry vaccines is that they have to cost about a penny a dose. Otherwise, it's not economically feasible. So you really have to get a very cheap vaccine. Fortunately, it isn't that hard to vaccinate poultry with an oil-based adjuvant, and you can actually mass vaccinate, 2-day-old chicks, but it has to be really cheap. And in the past, there were all these trade consequences. So the US and Canada didn't use vaccination, but they're now re-looking at it.
Great, thank you, Kanta. I mean, I think your question and the answer illustrates how the issues that we're facing go way beyond science. And we can have a scientific solution, but then there are other issues like trade that get in the way of actually implementing. Yeah. So it's a multifaceted problem.
Do you know at this stage whether the stem-specific antibodies are protective in animal models?
Yes, thank you. All right.
As a second,
Sure.
will you be testing in transmission models?
Yes, great, thank you. So we've managed to raise some money, some philanthropic money from the flu lab. Thank you very much, the flu lab. And so what we are hoping to do now is actually what we're setting out to do right now is to test the Rio H5 vaccine in lethal challenge studies with mice. And so we want to show that we can protect the mice against a lethal challenge with the H5, actually with the Texas virus strain. This is being done at Wisconsin in their influenza center. And protect the mice against H5, but we also want to see in that study whether we can protect against H1 with that vaccine, along the strategy that I outlined of trying to get protection against the seasonal, in addition to the seasonal flu vaccine. If that works, then our plans are to move on to the ferret model, where we can look not just at protection from lethal challenge in ferrets, but also ferrets are a very good, and in the field, somewhat reliable indicator of transmission, where you can put ferrets next to each other in separate cages and measure the transmission. And so we'll see whether or not we can reduce or prevent transmission in that model as well. But we're just now starting the mouse studies, so early days still, yes. Kanta, do you want to respond to that? Okay, yes.
Yeah, it was a little fuzzy on the antigen reorientation process.
Yes.
How that's fitting into the vaccination process. You know, you're spinning you're spinning these proteins. That's not going to last for a long time. So there's the point then to have them exposed long enough for the antibodies to start working?
Yeah, thank you for that question. When one formulates vaccines with alum, and in this case with the reoriented protein with alum, the protein binds very tightly to the alum. And that binding interaction is one that lasts probably for weeks in the body after it's been injected. And in fact, microscopy studies have shown that particles are broken off, and those particles contain small amounts of alum plus the protein. We haven't done it with our antigen, but with other antigens, it's been shown that those particles are taken up then by, taken up by what are called dendritic cells, which bring the particles to the lymph nodes, and it's been actually demonstrated that it brings them to the lymph nodes, and you can actually visualize this process. And so what we envision happening is that the particles, the alum plus the protein, will last long enough for this process to occur, and for the antigen to be presented to the immune system in the lymph nodes to allow for the elicitation of the antibodies. So yeah, the goal is to keep them together, and we have ways of measuring how tightly the protein binds to the alum, and we've shown that it's a very tight association in this case, right? Yeah.
Do you intend to use the reorganized HA vaccine in combination with the traditional head-based vaccines?
Yeah,
or?
thanks. Thanks for the question. The problem I have, I see, is that we're not going to be able to study protection against H5 in the absence of an outbreak, and we'd like to have this ready before an outbreak. And so what we're thinking is that, yes, we would study it on top of the existing seasonal vaccine and look for additional benefit against either H1 or H3 in that seasonal vaccine. It's a difficult study, and it's a high bar. I think following Jeffrey's great talk, one could consider doing a direct influenza challenge study instead, but logistically, I think that's going to be harder.
Yeah, I think what I worry about a bit is that when we do elicit stem-based immunity and you next encounter influenza again, the head wins again.
Right.
So maintaining the stem-based immunity on subsequent exposure, naturally or otherwise, will be challenging.
Yes. So what Kanta's talking about is that even if we elicit stem-binding antibodies in people, if they get exposed to H5N1, the response against the head is so strong that will be what the immune system kicks into high gear. What we're hoping is that because you already have memory B cells against the stem, that they as well would be stimulated, and while the anti-head response is being developed, the anti-stem response will already have kicked in from the memory cells. So that's the theory of the case, at least, right? Catherine.
Hi, Peter. I actually have a very related question, but in the opposite order. The immunization studies you've done have presumably been in naive animals, but I'm wondering about original antigenic sin and if they already have a head response, if you're still able to elicit those stem antibodies, if they've already seen flu.
Yeah, excellent question. So Catherine's referring to the fact that our response to a flu vaccine or to a flu infection is actually highly dependent on our previous history of flu vaccine or flu infection in what's been termed original antigenic sin or immune imprinting phenomenon. And it was most classically demonstrated with people that were alive during the 1918 pandemic and showing that their response to influenza vaccination was different than people that were not alive then, who were younger than that, because the people that were alive during the 1918 pandemic had their immune system primed in a different way than those that were younger. And so the concern here is that the mice that we're using are naive. They haven't been infected with influenza because they don't get infected with influenza. And so how do we know that our studies actually bear what's going to happen in humans? And the simple answer is we don't know. But the attempts to try and deal with that are we're immunizing animals with different strains of HA to immunize them, so to mimic prior exposure, and then see what happens when we come in with our vaccine, right? Flu is tough. Oh,
Peter,
Ann, yes.
Yeah. And I'm not sure I've got the biology correct, but so I understand that you've been able to do this flipping scheme to be able to generate an antibody response. The question is how well those antibodies will have accessibility to the stem
Yes.
in doing their job.
Yes, thank you. So this has been studied quite extensively by the antibodies against the stem that have been pulled out from humans that are rare but have been pulled out and characterized. And the answer is there's about a 2 or 3-fold decrease in the accessibility. And so the antibodies have a harder time coming in than for a freely exposed HA, but they still can get in with a 2 or 3-fold shift in their KD, in their affinity. We think the reason, or our hypothesis here, was that although the antibody can get in, maybe a big B cell, which is needed to get the whole process started, is going to have a much harder time getting in than a small antibody. And so therefore, our hypothesis was that if we flipped it upside down and made it more exposed, we could elicit the antibodies, and it looks like that's indeed what we get. Eliciting them, right.
And so you could take
You could take a hit, right, on binding, right. That's at least the hope. Yeah. Yes.
Thank you. You mentioned the response to people who were priorly exposed to influenza, to Spanish flu being different. There is a conflicting literature on other vaccines affecting vaccine response, like prior BCG vaccination. Can you comment on that? Are you looking at that interaction with other prior vaccines?
Yeah, thank you for that question. We're not at this point looking at that. The project is really too early for that, and I'm not an expert to talk about the potential implications of other vaccines. There are other people in this room who could answer that question better. Deb?
I think you made such a great point at the beginning about reassortment, and I think to date we've had this problem with the dairy workers in the summer and in the early fall, where we haven't had seasonal flu.
Right.
And with California persistent,
Right.
Texas has gotten pretty quiet, Iowa's gotten quiet, but with California
Just
still
started,
hot
right.
and within weeks of flu,
Right.
you can see we weren't at risk in the summer, but we now are approaching a very risky time for reassortment. And I
Yes.
think that's not really been taken into the context of
Yes.
why we've been talking about it and others haven't
Right.
in the media, but that's when the reassortment could be, particularly with asymptomatic spread, which
Right.
we're still not, we're not watching at all for avian flu asymptomatic spread. And so all it takes is an asymptomatic person to get a seasonal flu. And that's
Absolutely.
everything.
And there's been an uptick in the California H5N1 cases, so it is a concern. Thank you, Debbie. Yes.
That was a great talk. You've obviously had a lot of years in drug development. Given that our government, who's likely to be critical if and when a pandemic occurs, what do you think a realistic time frame is if we don't move everything forward and it's ready to go for actually putting this type of therapy or vaccine in practice? And could you contrast it to the mRNA vaccine
Yeah,
approach that we use in COVID?
Thank you for that question. I've been told by the experts that I've talked to that if there is a H5N1 pandemic outbreak, that it should be possible with current technology, and in particular, relying on the mRNA technology, as well as traditional technologies for developing flu vaccines, which uses a process that uses eggs, to come up with sufficient amounts of the vaccine to protect Americans in approximately 6 months. Is that okay? Is what the experts tell me. One complication is that if the bird flu wipes out egg production, then that's going to actually make one of those two avenues much harder. And there are certainly people and efforts to focus on that and to ensure that in the event that we have an outbreak, that hopefully will be ready within that 6-month period. There are, of course, two very serious issues with what I just said. One is that in the first 6 months, there's potentially a lot of people that will die. And unlike COVID, influenza kills not only the very old, but it also kills the young. And one of the saving graces of COVID was that it didn't kill the very young very much. The second issue with the statements that I just made is that pandemics are, of course, global. And so taking care of people in the U.S. in a 6-month period maybe is feasible, but taking care of the world is a whole nother issue. And I think that's a real concern. So the hope with this strategy, which, as I say, is orthogonal to what's currently being worked on, which is very important, the orthogonal approach here is to see if we can come up with something which would give pre-existing immunity, even if it's not great, to prevent severe disease and death. So if you get at least a little bit of immunity in vulnerable populations on the first day of the outbreak and not have to wait for 6 months.
That makes me think of whether to ask you whether you're looking at the neuraminidase as well.
Oh, yes. Lots of us are looking at neuraminidase, and that's certainly a very attractive target.
Yeah,
Yes.
because I think the current data suggests that the 2009 H1N1
Right.
neuraminidase has elicited some cross-reactive antibody to the avian H5N1.
Yes, God, I hope so. So for the non-experts, the H5, H refers to hemagglutinin, and the N1 refers to another protein on the surface, which is called neuraminidase. And there are different types of neuraminidase, but most of the human viruses are N1 or N2, so there are a couple subtypes. It turns out that in the traditional flu vaccine productions, the production of traditional flu vaccines, the neuraminidase component of the vaccine is actually damaged. It's pretty ineffective. And so the current vaccines don't give a very strong neuraminidase response, but natural infection does. And what Kant is referring to is that there's now some evidence that's starting to come out that suggests that people that actually were infected during the 2009 pandemic, which was an N1, are perhaps having immunity against the H5N1. And maybe that's why we haven't seen yet, hopefully won't, very severe cases of H5N1, right? And so the net result of that is many people and many companies are now starting to focus on the neuraminidase as a potential alternative strategy to develop influenza vaccines. Okay.
Just to follow up to that, so you mentioned people who were infected with H1N1. What about people who received the vaccine for H1N1 that was developed around 2009?
Yes, thank you. So it's possible that people who received the vaccine in the 2009 pandemic will also have some protection, but the actual way in which the vaccine is made involves a process because you need to make the vaccine non-infectious. So it involves a process in which the virus is inactivated. And it turns out that that inactivation process damages the neuraminidase protein. And so the immune response to the neuraminidase component of the vaccine is weaker than the neuraminidase response following natural infection. And so that's why there could be a difference, but we hope that people that got the vaccine will have some limited immunity, but I think it's jury's still out, yeah.
Thank you for that great talk. All the vaccines, to my knowledge, that are available now target our type 2 response, and yours does too. So you don't generate the type 1 response that creates a lot of T cell response. Can you talk about how your vaccine might be combined with other approaches that would stimulate stronger T cell response?
Yeah, thank you for that question. Protein vaccines such as this do not, in general, lead to very strong T cell responses, certainly not what are called CD8 positive T cell responses or killer T cell responses. One does elicit CD4 T cell responses that are helper responses. And I think that's a limitation of protein-based vaccines. The mRNA vaccines and certainly the live attenuated viral vaccines do a much better job of eliciting that. I think that's just a limitation of the system, right?
I think we have a break to digest all this delightful information. Please be back here at 11.